a zygote. In the 9 months, you were sitting in your mother's womb. You were being developed into a human being. Mouse, 21 days in mice, right? From zygote to the whole real animal that's out there. So please go read about it. I want you to know why I was saying it. Anyways, this was because this patterning of where things are in flies and mice and the screening, genetic screens for this actually discovered a lot of epigenetic genes. Okay? Because Hox gene expression, which dictates where things are going to be, completely depends on epigenome, just like any other gene. But Hox genes was obvious because in Drosophila, when they killed this gene called UBX, which is a homeobug gene, they started to make 4 fins rather than 2. Was a bit cool as somebody with 4 fins, right? It's cool. So it was bithorax. This phenotype is called bithorax because there are 2 of them right there. Okay. In the Muslim, you have same thing, house mutants. So mice, you see how the mutants have extra, I think, hands. You can have loss of function, you make 2 of them. You have gain of function, you don't make any. Isn't that cool? That's how a lot of people, you see abnormal people across, meaning abnormal means people who are defectively born. They're born because of defects in Hox genes for the most part, guys. Okay? All right? So you need to know when you see somebody who is not born in the correct way, you should question what genetic mutation this guy would have had. I find it super fascinating because to me it's like, whoa, nature error on the nature in a error in our Bhagwan, right? Our Bhagwan made an error. Nature made an error. A genetic mutation error was made because the chromosome was not segregated properly, or a mutation was not corrected by the DNA repair machinery. The guy was born, the kid was born with that. Got to have live like life with that. Down syndrome patients. How many of you seen Down syndrome patients? Right? You see, they all look alike, same, exact same. Only 1 chromosome. 1 chromosome. What happens in Down syndrome? Trisomy 21. Could I silence that extra 21 with epigenetic modifying things? Here is a question for you, man. You want to make $1 billion? You can make with that. Okay? There was a research earlier this year showing how CRISPR-Cas9 could be used to suppress trisomy 29 in a theoretical experimental world. That's a company idea right there. That'll make you $1 trillion. You wanna be rich? That's what you do. Okay? Idea. I'm giving you ideas. All right? You can do it. Somebody, Fang Zhang, made Jennifer Doudna made my CRISPR. I figured out how we worked on CRISPR. Somebody used it to kill trisomy 21's, effect, right? If I can have that embryo be, I would be saving so many people, so many things, right? Wouldn't that be fun? That's a far-fetched dream. It's very difficult to do, but you can do it. Okay? All right, guys. I'm going to try to end it here for you now before I'm gone for tea, but we'll talk about polycomb and trithorax. That's a classical epigenetic problem. Polycomb, trithorax. Polycomb does H3K27 methylation and suppresses genes at least transiently and in long term. So we talked about long-term silencing. We talked about transient silencing. Okay? All right, cool. We'll chat in a bit. So 15 minutes maybe. 10 to 15 minutes.